Welcome to the deep dive.
Okay, think about this for a second. What if some of the world's, biggest problems, things like climate change, maybe curing really tough diseases, or even dealing with all that plastic waste, what if they could be solved by something incredibly tiny, something you can't even see?
It sounds like science fiction, right?
Yeah.
But it's actually happening. It's becoming reality right now, really, thanks to this huge discovery in biology.
Exactly. So today we're going to do a deep dive into, well, the world of proteins, and specifically how artificial intelligence, AI, has cracked one of biology's oldest, most difficult mysteries, how proteins fold.
Yeah, and the scale is just wild. I mean, picture this. For over 60 years, you had tens of thousands of biologists really carefully mapping out maybe 150,000 protein structures. Hard, painstaking work.
Right.
But then in just a few years, this small team using AI, they figured out the structures of 200 million proteins. That's like almost every protein we know exists in nature. It's staggering. So our mission today is to unpack this. We'll talk about what proteins actually are, why their exact 3D shape is so critical for, well, everything living. We'll look at the massive challenge scientists faced for decades just trying to see these shapes. And then the really amazing part, how AI just completely changed the game.
And finally, what does this all mean? What are the implications for science, for medicine, for all of us?
Okay, let's jump in. Proteins, where do they even start?
Right. So at the most basic level, they start as simple chains. Imagine amino acids as like different colored beads. There are 20 different kinds.
Okay.
Each bead, each amino acid, has a standard part that lets it connect to the next one, but also a unique bit called a side chain. And that side chain gives each amino acid its own character, its own properties.
So the side chain is what makes each bead different. And these beads link up using something called peptide bonds, right?
Right, yeah.
Forming these really long chains.
Yeah, exactly. Long, kind of floppy chains initially. But here's the crucial part. They don't stay floppy. Because of all sorts of tiny forces between the atoms, like little magnets pulling and pushing, hydrogen bonds forming, how they react to water around them, these chains automatically fold up.
Spontaneously, just on their own.
Yeah, spontaneously into a very specific, stable, three-dimensional shape. And it's that final shape that dictates what the protein does. It's absolutely paramount.
So if the shape is even a tiny bit wrong, the protein just can't do its job.
Pretty much, yeah. Think of them as tiny machines built for one specific task, like hemoglobin in your blood. Its shape is perfect for grabbing onto oxygen molecules.
Okay.
Or the proteins in your muscles, they have shapes that let them slide past each other, causing contraction. It's all about the shape.
Makes sense. But you said figuring out these shapes.
Yeah.
That was the hard part.
Oh, incredibly hard. For a long, long time, determining just one protein structure was a massive, time-consuming effort.
So how did they even start? What was the first big breakthrough?
That came with a technique called X-ray crystallography. It's clever, but really difficult. Basically, you have to convince the protein molecules to line up perfectly and form a crystal.
Like a salt crystal, but made of protein.
Sort of, yeah. If you can grow a good crystal, you then shoot X-rays through it. The X-rays bounce off the atoms in a specific pattern, a diffraction pattern.
And that pattern tells you the shape.
Well, not directly. You get this complex pattern of dots, and then you have to do a lot of calculation working backward to figure out the arrangement of atoms that must have created that specific pattern. It's like figuring out the shape of a building by looking at how light diffracts around it.
Wow. Okay, sounds tough. Who figured this out first?
A British biochemist, John Kendrew. And it took him, get this, 12 years. 12 years just to map the structure of the very first protein, which was myoglobin.
Myoglobin, that's one in muscles, right? Stores oxygen.
Exactly. He actually started with horse heart, but the crystals weren't good enough, so he switched to whale meat from Peru.
Whale meat? Why whales?
Because whales dive deep, right? They need to store a lot of oxygen in their muscles, so they have tons of myoglobin, better chance of getting good crystals.
Smart.
Yeah.
So after 12 years, he finally saw it.
He finally got the diffraction pattern and worked out the structure. He actually said when he first saw the image, it was far more complicated than he had ever imagined. But yeah, he did it. Huge achievement. Got the Nobel Prize in Chemistry for it in 1962.
An amazing story.
Yeah.
But 12 years for one protein, that doesn't sound very practical for figuring out millions of them.
Not at all. That was the problem. It was groundbreaking, but excruciatingly slow and expensive too. Tens of thousands of dollars per protein, often.
Ouch.
And even after Kendrew's success, progress was slow. In the next 20 years, only about 100 more structures were figured out using crystallography.
So an entire PhD project might just solve one or two structures.
Easily, yeah. Growing the crystals is still a major bottleneck even today. It's part art, part science.
Okay, so crystallography was important, but clearly not the final answer. What was the alternative hope?
Well, scientists realized something else. Figuring out the sequence of amino acids in that initial chain. That's much, much easier and cheaper, like maybe $100.
Right, so the sequence is cheap, the structure is expensive and slow.
Exactly. So the dream, the sort of holy grail of biology for decades, became, could we just predict the 3D folded shape directly from the amino acid sequence? If you could do that, you'd save unbelievable amounts of time, effort, and money.
Were there any early signs this might even be possible?
There were hints. Linus Pauling, another giant in the field, actually predicted some basic repeating patterns in proteins, things called alpha helices and beta sheets, just based on the chemistry of the amino acids. That was one of biology's few real a priori predictions.
Okay, so predicting simple patterns was possible, but the whole complex 3D fold.
That seemed way harder. And this leads to something called Levinthal's paradox.
Paradox. Sounds intriguing.
Yeah. Cyrus Leventhal, a biologist at MIT, did a back-of-the-envelope calculation back in the 60s. He took a pretty short protein chain, maybe 35 amino acids long, and he calculated how many different ways that chain could theoretically fold. The number was astronomical, just unbelievably huge.
How huge are we talking?
Like more possible shapes than atoms in the universe by many orders of magnitude. He figured that even if a protein could try out billions of shapes per second, it would still take longer than the age of the universe to randomly find the right one.
Whoa.
Yeah.
Okay, but proteins do fold, and quickly.
Yeah.
In milliseconds sometimes.
Exactly. That's the paradox. They don't randomly search. They somehow find the right path, the right fold, almost instantly. But computationally, trying to simulate that search, it looked completely impossible, insurmountable.
So the prediction problem seemed doomed by sheer complexity, yet people didn't give up.
Nope. Scientists are persistent. In 1994, a professor named John Molt started a competition. The Critical Assessment of Protein Structure Prediction, or CASP.
CASP. Okay, what was the idea?
Simple, really. Every 2 years, CASP would release the amino acid sequences of proteins whose structures had just been solved experimentally, usually by crystallography, but hadn't been published yet.
So the answer was known, but secret.
Right. Teams from around the world would use their computer programs to predict the 3D structures from just the sequences. Then CASP would compare the predictions to the actual experimental structures and score them. A score of 90 or more was basically considered solved, getting it right.
How did the early years go? Were the predictions any good?
Not really initially. The first few CASP competitions, the best scores were like down in the 30s or low 40s.
Oof, that's not close.
No. There was apparently a lot of what they called foot tapping, a polite term for when scientists think someone else's prediction is just way off base. Friendly disagreement, you know?
Okay, but the competition must have spurred innovation, right?
Definitely. And it led to some really creative ideas. One of the leaders for a long time was a program called Rosetta, developed by David Baker's group at the University of Washington.
Rosetta? I think I've heard of that. Didn't they do something with, like, home computers?
Yes. That was Rosetta at Home. A brilliant idea. They basically turned protein folding prediction into a giant distributed computing project. People could download a screensaver, and when their computer was idle, it would use that spare processing power to help fold proteins.
Crowdsourcing computing power. Clever.
Right. But then something even stranger happened. People using Rosetta at Home started writing in saying, hey, I think I could fold this better myself than the algorithm is doing.
Wait, what? Regular people thought they could outfold the sophisticated computer program.
Yeah, which gave David Baker another idea. He thought, okay, what if we harness human intuition directly? So he created Fold It.
Fold It, the video game.
Exactly. It turned protein folding into a puzzle game. Players could literally grab parts of the protein chain on screen, twist them, turn them, trying to find the lowest energy, most stable fold, following some basic physics rules.
That's amazing. Did it work?
It worked unbelievably well.
Oh.
In one famous case, there was this enzyme from a virus similar to HIV that scientists had been stuck on for over a decade. They couldn't figure out its structure.
Wow.
They put it on Fold It. Within 3 weeks, about 50,000 gamers collaborating online solved it.
No way. Serious?
Seriously. Their solution was later confirmed by X-ray crystallography, and the gamers even got credited as co-authors on the scientific paper. It was this incredible demonstration that human spatial reasoning and intuition could solve problems that algorithms, at the time, couldn't crack.
That is just wild. It shows there's something about how we think that's different.
Absolutely. And one person who was really paying attention to Fold It was Demis Hassabis.
The founder of DeepMind, the AI company.
The very same. Former child chess prodigy, neuroscientist, AI guru, DeepMind was already famous for AlphaGo, the AI that beat the world champion Lee Sedol at the game Go.
Right. That was a huge moment for AI.
It was. And Hassabis, inspired partly by Fold It, wondered if AI could be trained to capture that kind of intuition, that pattern recognition, not just for games, but for fundamental scientific problems like protein folding.
So DeepMind decides to take on protein folding. Did they just jump into CASP?
Oh, pretty much. After AlphaGo's success, they held this internal hackathon. The challenge was, can AI learn to play Fold It, essentially? That led directly to the AlphaFold project.
And this was timely, right? Because you said the existing methods like Rosetta had kind of plateaued at CASP.
Exactly. The scores had stalled out. Even the best groups weren't making much progress anymore. Some were even getting slightly worse scores in subsequent CASPs. It seemed like maybe there was a fundamental limit to the older approaches.
So enter AlphaFold 1. What was their approach? Was it totally different?
Their first version, AlphaFold 1, used what was then a fairly standard deep neural network. They trained it on all the known protein structures in the public database, the Protein Data Bank.
Okay, learning from known examples. Makes sense.
But the key insight, the really clever bit for AlphaFold 1, was how heavily it relied on evolutionary information.
Evolutionary info. How does that help predict a fold?
Well, think about a protein like hemoglobin again. It has to work in humans, in dogs, in whales, across many different species. So its basic shape has to be conserved throughout evolution, even if the exact amino acid sequence changes a bit.
Okay. So by comparing the hemoglobin sequence from lots of different animals,
you can see which parts of the sequence don't change much. Those are probably crucial for the structure or function. But even more powerful is looking for coevolution.
Coevolution, like two things evolving together.
Precisely. Imagine you have two amino acids that are far apart in the linear chain, but in the final 3D fold, they need to be right next to each other. Maybe one is positively charged and one is negatively charged, holding that part of the protein together.
Okay.
Now, if one of them mutates, say the positive one changes, that might destabilize the protein, unless the negative one also mutates in a compensating way, maybe becoming less negative or shifting position slightly to maintain that crucial interaction.
Got it. So if you see two positions in the sequence that consistently mutate together across many species,
it's a very strong clue that those two amino acids are close together in the final 3D structure, even if they're hundreds of beads apart in the initial chain.
That's clever. Using evolution as a guide to spatial proximity.
Exactly. So AlphaFold 1 used these evolutionary comparisons to predict, essentially, a 2D map. This map, called a pair representation, showed the predicted distance and orientation between pairs of amino acids.
Like a probability map of who's next to whom.
Sort of, yeah. And then a separate algorithm took that 2D map and used it as a set of constraints to actually fold the 3D chain.
Okay, 2 steps. Predict the 2D relationships, then fold in 3D based on that map. How did it do at CASP? This was CSP 13.
Yes, CASP 13. And AlphaFold 1 was the clear winner. Big improvement over previous methods. But its score was around 70.
Better, but not the 90-plus solved benchmark.
Right. It was a major step forward, proved the power of deep learning and evolutionary data, but it hadn't cracked the problem. DeepMind knew they were onto something big, but they also knew they needed something more. They had to go back to the drawing board.
So, promising, but not the breakthrough yet. There must have been some pressure. Did they think maybe the problem was just too hard, even for their AI?
Maybe briefly, but Hassabis basically tasked John Jumper, who had been a key researcher on AF1, with leading a new effort. The mission was clear. Solve it. Don't just improve, solve it.
John Jumper takes the lead, and what they came up with next, AlphaFold 2. That was the one that changed everything, right?
That was the earthquake. Yeah, AlphaFold 2 was a fundamental redesign. Its core innovation was incredibly ambitious. They integrated the geometric reasoning, the physical constraints, and the evolutionary insights all directly into the deep learning network itself. It wasn't separate steps anymore.
Wow. Okay, how did they pull that off? What were the key ingredients?
Well, people often point to three things, though one is a bit of a surprise. First, massive computing power. Being part of Google gave DeepMind access to enormous resources, especially Google's specialized AI chips, the tensor processing units, or TPUs. That definitely helped.
Sure, brute force computing helps.
Yeah.
What else?
Second, large data sets. They trained on the protein data bank, all those known structures. But here's the kicker.
Yeah.
They used the exact same training data for AlphaFold 2 as they did for AlphaFold 1.
Wait, really? The data didn't change?
Nope, which powerfully demonstrated that the algorithm was the main improvement, not just throwing more data at it. The quality of the AI thinking mattered more than the quantity of examples.
Okay, so that brings us to the third pillar, the crucial one.
Yeah.
Better AI algorithms. What was different this time?
They drew inspiration from a cutting-edge AI architecture called the Transformer. This is the tech that powers things like ChatGPT.
Ah, the T in ChatGPT. Okay, what's special about transformers?
One key idea is attention. It allows the AI to weigh the importance of different parts of the input data when making a prediction. Like in a sentence, understanding which pronoun refers to which noun based on the context.
Right, like your example earlier. The animal didn't cross because it was tired. Attention helps figure out it is the animal.
Exactly. AlphaFold needed something similar for amino acid sequences. Which part of the sequence influences the fold of another part, even if they're far apart? So they built a custom version, a heavily modified transformer architecture they called the evoformer.
Evoformer, okay. How did it work?
This was the radical part. Instead of the two-step process of AF1, the evoformer had like two interconnected towers or processing streams operating in parallel. One focused on the raw sequence and evolutionary data, the biological information.
The biology tower.
The other focused on the geometric relationships between amino acids, the pair representation map, but now much more sophisticated, the geometry tower.
And these towers work together?
Yes, constantly. That was the breakthrough. They didn't just run separately. There was this complex bridge allowing information to flow back and forth between them multiple times.
How did that help?
Well, the biology tower would process the evolutionary data, find those conserved sequences, find the co-evolving pairs we talked about, and it would send clues about likely spatial arrangements over to the geometry tower. The geometry tower would then use its attention mechanisms, including a clever trick called triangular attention, looking at relationships between triplets of amino acids to update its map of distances and orientations. It would figure out, okay, if A is close to B and B is close to C, then A must be this far from C. Basic geometry, but applied across the whole protein structure.
Using the triangle inequality, basically.
Exactly. But then, crucially, the geometry tower would send its refined structural understanding back to the biology tower. It might say, hey, this evolutionary connection you found, it implies a physically impossible structure, so maybe don't weigh that connection so heavily.
So they're cross-checking each other. Biology informs geometry, geometry informs biology.
Constantly. This exchange, this iterative refinement, happened something like 48 times within the evoformer network for a single prediction. It allowed the network to build this incredibly rich, internally consistent understanding of both the biological constraints and the physical possibilities.
That sounds computationally intense, but incredibly powerful. Was that the whole story of AlphaFold 2?
That was the evoformer, the first major innovation. The second was the structure module. This is the part that actually built the 3D protein model.
Okay, how did that work differently?
Instead of just using the final 2D map as a constraint, the structure module worked more directly in 3D space. For each amino acid, it defined a sort of local coordinate system, a frame, based on 3 key atoms.
A frame for each amino acid.
Yeah. And the network's job was then to predict how to move and rotate each of these individual frames from a starting point, like the origin, to their correct final positions and orientations in 3D space.
So it's predicting the exact 3D coordinates for each amino acid's frame.
Essentially, yes. But here's another really unique kind of counterintuitive insight they had.
Oh.
Initially, the structure module didn't explicitly enforce the fact that the amino acids are connected in a chain.
Wait, it ignored the chain. How can it fold a protein if it doesn't know it's a chain?
It's like they gave the module a bag of amino acids. It learned to place each amino acid frame independently in 3D space first, focusing just on getting the local structure and the relationships predicted by the evoformer correct.
A bag of amino acids.
Nice.
That sounds really weird. Why do it that way?
The thinking was that enforcing the chain constraint too early could trap the prediction in a wrong fold. By letting the amino acids find their preferred local environments first, almost ignoring the backbone connections initially, it could explore the solution space more effectively and avoid getting stuck.
So it places the pieces roughly where they should be, then worries about connecting them properly.
Kind of. That's why if you watch the visualizations of AlphaFold 2 folding a protein, sometimes it looks physically impossible in the intermediate steps chains passing through each other, weird knots forming before it suddenly snaps into the correct, final, physically plausible structure.
Fascinating. So, bag of amino acids, place them, then refine.
And refine again. The initial 3D structure produced by this module wasn't even the final answer. They actually fed that predicted 3D structure back through the evoformer network several more times.
Looping back again, why?
To allow the network to reason even more deeply about the relationships, now with a concrete 3D context. It's like showing it its first draft and asking, okay, now that you see this shape, can you refine your understanding of the evolutionary and geometric connections even further?
Wow. So multiple passes through evoformer, structure module with bag of acids trick, then more passes through evoformer. It's an incredibly sophisticated process.
Absolutely. A true end-to-end system where every part informs every other part.
And the result, CSP14, December 2020, what happened?
The result was triumph. Absolute triumph. AlphaFold 2 didn't just beat the competition, it essentially solved the problem. Its predictions were, on average, achieving scores well above 90. They were often indistinguishable from the experimental structures determined by X-ray crystallography within the limits of experimental error.
Indistinguishable from experiment. That's the gold standard.
That's the gold standard. John Molt, the CAFP organizer, sent this email basically saying, you guys nailed it. John Jumper, who led the AF2 team, described it as this moment of sheer excitement, realizing they'd finally done it after years of intense effort.
I can only imagine. And the scale we talked about earlier.
Yeah.
60 years for 150,000 structures by the whole world.
And then AlphaFold 2 comes along, they put it to work, and very quickly they predict structures for over 200 million proteins. From humans, mice, bacteria, plants, pretty much the entire known proteome of life on Earth.
It's like going from mapping a few cities by hand to having Google Maps for the entire planet overnight.
That's a great analogy. It compressed decades, maybe even centuries, of future experimental work into just a few months. It just handed the biological community this enormous treasure trove of structural information.
So what has been the impact? What are scientists doing with this treasure trove?
Oh, the impact is already profound and widespread. Immediately, researchers started using the predicted structures. People working on, say, a malaria vaccine could suddenly see the shape of key parasite proteins they needed to target.
Okay, vaccine development. What else?
Understanding antibiotic resistance. Bacteria evolve enzymes that break down our antibiotics. AlphaFold provided structures for many of these enzymes, helping scientists figure out how to block them, maybe make old antibiotics effective again.
That's huge.
Yeah. Understanding genetic diseases. Many diseases, like certain cancers or even conditions like schizophrenia, are linked to tiny mutations in proteins. Now, researchers can see how those mutations might change the protein's shape and disrupt its function.
So connecting genetic code to actual disease mechanisms through the protein structure.
Exactly. And it's not just human health. Biologists studying rare or endangered species where it's impossible to do large-scale experiments suddenly have access to predicted structures for their proteins. It helps understand their unique biology and potentially aid conservation efforts.
The breadth is amazing. And you mentioned the AlphaFold 2 paper. Over 30,000 citations already.
Yeah, it's one of the most highly cited papers in recent memory, which just shows how fundamental this breakthrough is across all of biology, and of course, the Nobel Prize.
Right. The 2024 Nobel Prize in Chemistry. Half to John Jumper and Demis Hassabis for AlphaFold. Who got the other half?
The other half went to David Baker, the Rosetta and Folda guy we mentioned earlier, but interestingly, not for his prediction work.
No. What for then?
For taking the next giant leap. Not just predicting existing protein structures, but designing completely new proteins from scratch.
Whoa. designing proteins that never existed in nature.
Exactly. Solving what's called the inverse folding problem. Instead of sequence to structure, it's more like function to sequence and structure. Telling the computer, I need a protein that does this specific job, and having it design a brand new one.
How does he do that? Is it AI again?
Yes, it's generative AI.
And
His lab developed a technique called RF diffusion. It's actually conceptually similar to AI image generators like DALL-E or Midjourney.
how so? How do you generate a protein like an image?
Well, image generators learn by taking noisy images and learning to remove the noise to create a clear picture. RF diffusion does something similar with protein structures. It learns the rules of stable protein folds by looking at millions of examples, adding noise, messing them up slightly, and learning how to put them back together correctly.
Okay, it learns the principles of protein structure.
Right. And once it learns those principles, you can give it a prompt. Not a text prompt like a cat riding a bicycle, but a functional prompt. Like, I need a protein that binds tightly to this specific molecule, or I need an enzyme that performs this chemical reaction. And RF diffusion can generate computationally completely novel protein structures designed to do that job.
That sounds like a superpower for biology. Can you give an example?
A huge one they're working on is snake antivenom.
Antivenom. How does protein design help there?
Well, traditional antivenom is made by injecting small amounts of venom into animals like horses or sheep, and then collecting the antibodies their immune systems produce.
Right, I knew that.
But the problem is, those are animal antibodies. When injected into humans, they often cause severe allergic reactions, sometimes even fatal ones. It's a big issue, especially in poorer regions where snakebite is common.
Okay, so how does design protein help?
Baker's lab used RF diffusion to design completely synthetic, human-compatible protein binders, essentially artificial antibodies that specifically target and neutralize the most dangerous toxins in deadly snake venoms, like from the black mamba.
Human-compatible binders designed on a computer.
Exactly. These aren't derived from animals at all. They can be produced cheaply and stably in large quantities using standard biotech methods like brewing beer. And because they're designed to be human-like, the risk of allergic reaction should be vastly reduced.
That could save hundreds of thousands of lives, right?
Yeah.
Especially in places without good refrigeration or healthcare.
That's the hope. It's potentially game-changing for a massive global health problem, and it's just one example of what protein design is starting to make possible.
So beyond antivenom, what are scientists most excited about for the future of this protein AI?
Oh, the list is long. Definitely more powerful and precisely targeted vaccines are high on the list.
Okay.
There are already designed proteins entering human clinical trials for treating cancer and autoimmune diseases, proteins designed to specifically modulate the immune system in new ways.
Wow, clinical trials already.
Yeah. And then there's the environmental side. People are designing enzymes, which are just proteins that speed up chemical reactions to do amazing things like capture greenhouse gases directly from the air, maybe fix methane, or enzymes designed to break down plastics much more efficiently than anything nature evolved.
Enzymes to eat plastic or capture carbon. That sounds revolutionary.
It really is, and the speed is accelerating too. Baker calls it cowboy biochemistry.
Cowboy biochemistry, I like that.
Yeah, because instead of years of painstaking lab work, you can now design a protein on your computer, send the sequence off to a company, and have the actual physical protein delivered back to you in maybe just a couple of days ready for testing.
Design on Monday, test on Wednesday. That completely changes the pace of discovery.
Totally. It makes biology feel much more like engineering. You can design, build, test, iterate incredibly quickly.
And you mentioned this isn't just limited to biology, right? AI is tackling fundamental problems elsewhere, too.
Absolutely. What AI did for protein folding is a template. DeepMind, for example, has another program called Genome, focused on material science.
Genome, what did it find?
Using similar AI principles, Genome analyzed known crystal structures and predicted 2.2 million new, potentially stable crystalline materials. That's nearly 10 times the number previously known.
2.2 million new materials. What could they be used for?
Well, among those, they identified over 400,000 materials predicted to be highly stable, the kinds that might actually be synthesizable and useful. Things that could lead to better batteries, more efficient solar cells, perhaps even new superconductors. It basically jump-started decades of materials discovery.
So AI isn't just solving one big problem like protein folding. It's finding ways to solve these fundamental root problems across different scientific fields.
Exactly. These are problems where understanding the basic principles allows you to unlock progress across a huge range of applications. Cracking the code of protein folding accelerates drug discovery, vaccine development, enzyme design. Cracking the code of stable materials accelerates energy technology, electronics, construction.
It feels like we're entering an era where the rate of scientific discovery itself is accelerating massively.
That's the key takeaway, I think. Human knowledge is expanding faster than ever. Doubling the speed of discovery is great, but speeding it up by factors of hundreds or thousands, that changes the entire landscape. You start asking completely different questions, attempting things that were simply unthinkable before.
And the knowledge gained from things like AlphaFold or GenoA,
Yeah.
it's permanent, right?
Yeah.
Even if AI development stalled tomorrow, we still have those 200 million protein structures, those 2 million material candidates.
Precisely. These breakthroughs are generating foundational knowledge and tools that will benefit science and humanity for decades, regardless of what happens next with AI itself. The discoveries are real and lasting.
It really is mind-boggling. AI has basically transformed our understanding of life at its most fundamental level, the protein level. We've gone from decades of slow, careful structure mapping to this sudden flood of information, and now even the ability to design brand new molecular machines.
Yeah. Moving from painstaking discovery to rapid creation, it's a paradigm shift.
It opens up possibilities that, honestly, sounded like pure fantasy just a few years ago.
Yes.
Curing intractable diseases, designing materials atom by atom, maybe even cleaning up the environment with custom enzymes. It sounds like an absolutely amazing future. As long as, the AI doesn't get too smart and decide to destroy us all first.
Well, that's a thought for another deep dive, perhaps. But seriously, the potential upside here is immense. It's worth thinking about how this acceleration in fundamental science could ripple outwards and impact, well, pretty much everything.
Absolutely. A lot to ponder there. Thanks for unpacking that incredible story.
My pleasure. It's truly a fascinating time in science.